Hi there, welcome to the online platform of learning by the Department of Biotechnology. Continuing to my previous videos on antigen antibody interactions where we use it for the diagnosis purpose. In this video, we will be talking on going beyond those diagnoses for the analytical purpose of the antigen antibody interaction. Starting with immunofluorescence. Immunofluorescence is a technique or a method by which we try to identify whether there is the presence of a particular protein in question in the cell or in the tissue region. Immunofluorescence is done basically by two ways. Number one, by direct method or the indirect method. In the direct method, we take an antibody, a monoclonal antibody tagged with a fluorescent, and we incubate this fluorescent tagged antibody with the cellular system, believing that this cellular system is expressing the protein in question. If the protein in question is expressed, then the fluorescent tagged antibody will bind to it and will not be washed away during the washing process. On subjecting a particular wavelength of light, fluorescence can be seen if the protein in question is available. If the protein in question is not expressed by the cell, then there will be no binding between the protein and the fluorescent tag antibody, and there will be absence of fluorescence. The another way is the indirect method, in which we use two antibodies. First one is an untagged primary antibody and the second one is the fluorescent tagged secondary antibody. Again, we take the cellular system and incubate it with the primary antibody, believing that the protein in question is expressed. If the protein in question is expressed and is present over the cellular system, then the primary antibody will bind to it, and on incubating it with the fluorescent tagged secondary antibody, the secondary antibody will bind to the primary antibody, and on the subjection of the particular wavelength of light, fluorescence can be seen. The indirect method is only being done to increase the intensity of the fluorescence. Now, in the direct method, we are using the monoclonal antibody, and so a single protein can be bound by a single antibody. But when we are talking about the indirect method, the secondary antibody being polyclonal in nature, and it more than one secondary antibody will bind to a single primary antibody, and hence increasing the fluorescence intensity. So immunofluorescence is basically used to identify the presence of a protein in the cellular system. This could be used to know whether a cell is expressing the gene that we have inserted. We can even use it to know that whether a particular protein is expressed in a given condition, like in cancer biology. The next advanced assay is called as immunoelectron microscopy. Now, immunoelectron microscopy is a combination of immunoassay with transmission electron microscope, which is used for localizing the antigen over the surface of a cell or a viral particle. In this method, we take tagged antibody, and we incubate with the specimen of choice, and then transmission electron microscopy is being done. As you see in the picture below, the black dots are the areas where the antibody has bound to it, bound to the antigen. Now, these regions shows the presence of the antigen towards which the antibody has been generated. This gives us the idea where the antigen is present over the structure of a cell or the structure of a virus. This gives us a choice of developing drugs towards these particular regions. So, immunoelectron microscopy is basically done to understand the positioning of the antigen over the surface of a cell or a virus. The next important assay is called as surface plasmon resonance. Surface plasmon resonance has the principles of physics in it, much in it. But we will be looking it in a very simple form how surface plasmon resonance is basically used in immunological assays. Surface plasmon resonance, or abbreviated as SPR, is basically used for the characterization of ligand binding. And here we use SPR for understanding the affinity of an antibody towards a particular antigen. In this method, what we use is a metal film, a prism, and a light source. Now, what we know of surface plasmon is a phenomena that arises due to the oscillation of the free electron, and the resonance is generated through the light. Now, what we know is SPR is basically seen when a polarized light is subjected to a metal film, and then there will be a change in the oscillation of the free electron in lieu with the resonating light because of the difference in the refractive indices. Now, what we know is upon binding, there will always be change in the oxidation of the electron as compared to the free state and the bound state. Now, in this method, what we do is we take a gold film and we immobilize the antigen over it. When we do so, you can simply see that before the binding, there will be a different SPR reading as compared to the SPR reading which is being projected on binding with the antigen. And when we add the antibody, there keeps on changing the reading of the SPR. Now the change in the reading of the SPR is basically calculated through a method called as Freshman configuration, which has a deep physics principles of physics within it, and we'll not be talking much about it. But what we can understand in this method is when a particular wavelength of light is being subjected, and if there is a binding occurs, then there is a change in the intensity of the light that is reflected from the surface, or we can say in other words there will be change in the surface plasmon. Now the change in the surface plasmon shows a dark region over the photographic plates. Now these dark lines or dark regions give us a lot of information. As you can see in the graph that initially when there was no binding, there was when there was no binding, there was no change in the value of resonance, and the no change in the value of resonance indicated that there is an absence of binding. Then you see that there is a steep exponential change in the value of resonance upon when the antigen and antibody binds. So when an antigen and antibody binds, on that binding what happens, there is a change in the reflection intensity which is being calculated by Freshman configuration and can be seen over the graph. And again, when through washing the complex is dissociated, again there is a decrease in the value of the SPR, or there is a change in the intensity of the reflection. So these changes in the intensity of the reflection helps us to understand whether the antigen and antibody are binding, what is the affinity or the degree of binding between the antigen and antibody is present. So SPR is a very handy tool in understanding whether an antigen and antibody are interacting with each other, and what is the degree of affinity between the two. The last assay that we will be talking is on cell cytotoxicity assay. Cell cytotoxicity assay is being done to understand whether a effector immune cell is killing the target cell or not. the assay that there are many assays available for identifying the CCA. The one that we will be talking of is chromium 51 release assay. Now, when we talk about the chromium 51 release assay, this is the very initial, one of the very initial method that was being utilized by Doherty and Zickernagel. Now, in this method, what we do is we take the target cell and we incubate it with chromium 51. The target cell accumulates chromium 51. The accumulated chromium 51 target cell is then incubated with the effector immune cell. If the effector immune cell identifies a target cell, then it will go on killing the target cell, which will cause the chromium 51 to be released in the surrounding medium. The identification of the presence of chromium 51 after the incubation of the effector cell with the target cell give us the information that whether the effector cell is acting against the target cell or not. If there is an absence of chromium 51 after incubation, then it tells a story that the effector cell is not binding with the target cell and there is an absence of cell cytotoxicity. So, these were some of the advanced assays that are utilized in the immunological research to identify or to get number of information for analytical purposes. So, thank you for watching this video, and if you have any question, any queries or doubt regarding this essays that we have talked about, you are free to contact me through the contact details that which you see over the screen. Thank you for listening us, and we'll continue to talk on more such essays in my next video. Thank you.